Ready to unravel a mystery. Everyone knows Watson and Crick cracked the code of DNA, but honestly, there's way more to the story.
It's like a detective novel, right? You got to follow the trail of clues and the brilliant minds who found them.
Exactly. And that's our deep dive today, going beyond the textbook version. We're digging into an excerpt from Nature Education, Discovery of DNA Double Helix, Watson and Crick. Get this. Our story starts way back in 1869.
Whoa, 1869, that's like before the internet, before cars.
Before Coca-Cola even.
Right, ancient history. But seriously, that's when this Swiss chemist Friedrich Miescher stumbles onto something huge.
The excerpt calls it a remarkable discovery. He's studying white blood cells, trying to figure out what they're made of.
Which makes sense, basic biology building blocks.
Exactly. But he keeps finding this weird substance, not protein, which is what he expected. This stuff had tons of phosphorus.
And that's the chemist in him. Miescher, he's not just a biologist, so those odd chemical properties, they set off alarm bells. He even thought, hey, maybe this is something totally new.
No one was thinking about DNA like that back then.
Yeah.
But here's the sad part. The excerpt says his discovery was basically ignored for decades.
Tragic, right, like finding a hidden door but not opening it. Even Erwin Chargaff, we'll get to him later, he pointed out how some big science history book completely left Miescher out.
Talk about not getting your due. It makes you wonder how much groundbreaking stuff is just sitting there waiting to be rediscovered. But okay, so we've got this forgotten genius and his mystery substance. What did he even call it?
Oh, he called it nuclein. Catchy, right?
Yeah, nuclein. Sounds kind of ominous, but okay. So the story's got to pick back up.
Oh.
Right, we're on the trail of DNA.
We are. Fast forward now to the early 1900s, we meet Phoebus Levine. This guy was obsessed with the chemistry of life, published like hundreds of research papers on it.
Talk about dedication. What was Levine's deal? What was he after?
He was determined to crack the code of Miescher's nuclein, and he's the one who figures out it's made up of these repeating units, nucleotides. Each one had a sugar, a phosphate, and one of 4 bases: adenine, guanine, cytosine, and thymine.
Wait, those are the famous A, T, C, and Gs of DNA.
Bingo. Levine basically gave us the DNA alphabet right there. He even figured out they link up in a chain, the sugar-phosphate backbone.
So Levine lays the groundwork, but I'm guessing it wasn't just stringing those nucleotides in a line.
You got it. Levine thought it was a simple repeating chain, all 4 bases in equal amounts. Called it the tetranucleotide hypothesis. Good start, but way too tidy, as you'll see.
Ooh, okay, so what's the missing link? How do we get from a straight line to the elegant double helix?
Well, that's where our next unsung hero enters the scene, Erwin Chargaff. And his story, it's one for the books. Chargaff really shakes things up. It's the 1940s, and his research throws a wrench into Levine's nice, neat model.
Really? So what did he find? Another happy accident like Miescher?
Not quite an accident, more like serious observation paying off. Chargaff was studying DNA from all kinds of things, bacteria, humans, you name it. And he noticed something about those 4 bases, the A, T, C, and G.
They weren't showing up in equal amounts like Levine thought.
Exactly. Different species had different ratios, like a different recipe for each organism's DNA.
Okay, makes sense. More complex, but it fits, right? Every species is unique.
Exactly. Suddenly, DNA wasn't so simple. It was way more diverse, more potential for information than anyone thought. But hold on, Chargaff keeps digging, and he finds another pattern, what we now call Chargaff's rule.
Okay, yeah, Chargaff's rule. Refresh my memory, what's so special about it?
He found that no matter what the species, the amount of adenine always matched thymine, and guanine always matched cytosine, AATGC.
Those ratios, that's a huge EE clue about DNA structure. Even if Chargaff didn't quite know what it meant yet, it's like built-in instructions for copying itself.
You're getting it. But even with Chargaff's rule, it's still early days. It's the 1950s, and the double helix is still hidden.
So how do we get there? From ratios and clues to that elegant structure.
That's where things get really interesting. Because now, Watson and Crick enter the scene. But remember, they weren't starting from scratch.
Right. Miescher, Levine, Chargaff, they laid the groundwork.
Exactly. And they get a boost from a newish technology at the time, X-ray crystallography. Basically, you shine X-rays at molecules to see their structure.
Those X-ray images always look so messy to me. How could anyone make sense of them?
That's where Rosalind Franklin and Maurice Wilkins come in. They were the X-ray experts. Think of it like taking shadows of an object from all different angles.
Oh, and you piece together the shape from the overlapping shadows.
Exactly. So Watson and Crick have these shadow pictures of DNA from Franklin and Wilkins. They've got Chargaff's rule hinting at base pairing, and they're using, wait for it, cardboard cutouts.
Cardboard cutouts? Seriously?
Seriously. Building models, trying to make all this information fit. And it's during one of these sessions that Watson has his famous eureka moment.
Okay, now I have to hear this. Was there a light bulb? Did he jump up shouting?
Almost. He realizes that if he pairs adenine with thymine and guanine with cytosine, not only does the chemistry work perfectly,
But the pairs are the same size and shape, fitting into the X-ray data.
you got it, like a lock and key, but for DNA. It's not just pretty, it makes chemical and structural sense.
Whoa. So what does this mean? What does this discovery do for our understanding of DNA?
It changes everything. This double helix, with its base pairs, it's the perfect way for DNA to copy itself. Each strand a template for a new one. Elegant, simple, and it explains heredity at the most fundamental level. One of biology's biggest mysteries solved.
So we've got our elegant double helix, A with T, C with G, like some kind of microscopic puzzle snapping into place. It feels like the end of the story, right? DNA structure solved.
It's beautiful, no doubt. But remember, science is rarely a straight line. Turns out there's more to the story. Even with this structure, there are still some twists and turns.
Wait, really? More to uncover? I thought the double helix was the final word on DNA.
The double helix is the foundation. But remember, the excerpts mention different forms of DNA. We've been talking about bDNA, the classic one. But there's also aDNA, and get this, zDNA.
Okay, hold on. Different forms, like water can be ice or steam, but it's still H2O.
Perfect analogy. Same DNA, but the shape, the twist, it can change. Depends on things like what's around it, even the sequence of those bases. aDNA, for example, it's kind of like the compact version, squished a bit compared to bDNA, but then
zDNA.
zDNA. Okay, now that just sounds mysterious. What makes it so different?
picture a spiral staircase, right? bDNA, that's your typical staircase, twisting to the right. zDNA, that was twisting to the left. A left-handed helix.
Whoa, a lefty. Okay, that's cool and all, but what does it even mean? Do these different forms actually do anything different?
That's the million-dollar question. We're still figuring it out, to be honest. But there's more and more evidence that they're not just, like, oddities. They matter. For example, zBNA seems to pop up when DNA is being read to make proteins, maybe even involved in replicating itself. Some research even suggests it helps fight off viruses.
No way. So DNA is like way more complex, way more dynamic than I ever learned back in school. From a forgotten discovery in 1869 to a double helix, and now it's like